Hi, my name is Chloe Blunk, and this is for week 7 lab for Los Angeles Pacific University. In this lab was just antibiotic sensitivity with the Kirby Bauer diffusion test. So I started the lab on Thursday. I had to recreate an agar plate, and with the giant plate, I had to put it in 3 different sections: one for the penicillin, one for novobiacin, and the last one for so hopefully I pronounce this right. The gentamicin. My hypothesis was that penicillin was going to be the most efficient antibiotic, only because we know with research that it's the most commonly used because it focuses on gram-positive and with some gram-negative cells, but that's the most common antibiotic that people will use. That's most that works the best. So I checked my experiment. I started on Thursday. Today is now Sunday. I had checked it yesterday. Saturday was the 18th. Didn't find very much growth on it. Just a very, very small, like thin, like film kind of that was very difficult to see. The picture I took for my lab report was very hard to see as well. I had to put it under a very dark colored. I did a brown background to try to show it on the image. I did see where the area inhibition occurred, which is basically where the antibiotic worked with killing the bacteria. You can see like a little circle around each disc. My results for penicillin was 150 milliliters in not diameter, but with halfway from the center of the disc to the edge of the circle. So if you times that by 2, the whole entire circle was 300 millimeters in diameter. The novobiacin came second, and that was going to be 10 milliliters across the entire circle. And then gentamicin was the smallest one, and it was about total 1 millimeter across, showing that penicillin was indeed the most effective. Gentamicin, sorry, I hope I pronounce it right. Gentamicin was the least effective because it focuses on gram-negative bacteria, and S. epidermidis is gram-positive, so it shows that it was on the bottom of the scale of most effective. And then just a difference with some of the questions that were on here with between a bactericidal and bacteriostatic antibiotics. Bactericidal, it causes cell death, and it's gram-positive targeted, whereas the bacteriostatic antibiotics target DNA gyrase, and it causes an inhibition and growth. So it doesn't kill cells, it just stops the cells from growing. So that's basically it for my experiment for this week, for week 7, and it's the last experiment of the course. I really enjoyed all the labs and getting to clean out my fridge of all the live cultures. And that completes my microbiology class for the labs at Los Angeles Pacific University. Thanks.